Stem cells are amazing cells. They are a very small number of cells in our bodies that have two special properties. The first one is they can self-renew, namely whenever they divide, they can make a copy of themselves. So they become so-called immortal cells during our lifespan. And the second property is that they can generate many specialized cells or ordinary cells that are directly involved in tissue functions. And that's a process called differentiation. So because of these two key properties, that makes stem cells very special. My lab has worked on lots of things, but the common theme is working on the key genes that regulate the self-renewing ability of stem cells. And particularly, we focus on those genes which are not traditionally studied. Genetic information encoded in our DNA is then made into copies of blueprints called messenger RNA, guide the production of proteins. And this supposedly leads to the start of entire life process. But actually, recently, we found that the story is not that simple, because protein coding genes, they are not the entire picture of the secret of life. And they actually only occupies 1% of our genome. How about the other 99% of DNA? People used to, show no function in the DNAs and call them junk DNAs. And recently, we discovered that so-called junk DNA are hiding different kinds of genes, and they're equally important. We have 23,000 genes in our human genome, but we have 1 million of jumping genes in the junk DNA. Now we found that they have very important function in regulating the protein coding genes. So we take a very systematic approach, namely that's a multidisciplinary approach from a methodology perspective. First, we usually use cell or developmental biology methods like microscopy, transplantation, or laser ablation to identify stem cells in tissues. And once we know where are the stem cells exactly located in the tissues, then we take a genetic approach to knock out genes in the genome to see which genes knockout will lead to stem cell defect. And then after that, we take a molecular, biochemical, and also genomic approach to study how the gene looks like, what kind of function they will do when they make their proteins, and what will their protein will be doing in the cell to make certain kind of stem cell structure or turn on or off certain signaling pathways in stem cells. Recently, my lab also took on structural biology approach. We are using cryo-electron microscopy to study those key proteins that determine stem cell fate, how they look like, and how their function is moved in 3D and 4D way to really switch the function of other molecules in the cells. In my lab, we have been focusing on when these stem cell genes are active in non-stem cells, or when they are overly active in stem cells, what kind of cancer they will cause. And we found that many types of cancers are actually related to this. And so this is very exciting for us because it provides a new approach to understand and potentially to treat cancers. Now we can use small RNAs that specifically knock down these overly active stem cell genes, and we see tremendous impact. And so this is a method very different from the current immunotherapy. We hope to actually develop or help develop a completely new type of targeted approach to cancer research.